Ladies and gentlemen, on behalf of BB Biotech, I would like to welcome you to our virtual Bio Day 2020. Thank you very much for connecting, for your interest in BB Biotech, and for staying with us for the next 90 minutes. Innovation, genius, and perseverance are key for the biotech industry and its progress. If we succeed in eliminating the corona pandemic permanently, it will become clear that medical innovation has great social, economic, and political relevance. Both the health and the biotech sector will benefit from this. Dr. Daniel Koller will illustrate drivers within the current BB Biotech portfolio using examples of next-generation rational drug design, genetic medicine, and mRNA therapies. Just about 11 months after the publication of the genetic sequence of the coronavirus, vaccine products could impact the dynamic of the pandemic using a breakthrough technology that could change the way we fight disease and illness in the future. We will hear more on mRNA technology directly from Mr. Stéphane Bancel, CEO of our portfolio company, Moderna. A last note before we start. At the end, we will entertain a Q&A session. Please start sending your questions for our speakers already during the presentations. A selection can be made like that. Thank you very much. With that said, I'm very happy to introduce Dr. Daniel Koller, head of the management team of BB Biotech.
Thank you for attending BB Biotech's virtual Bio Day and a welcome from the whole team. It is my pleasure to give a quick overview of BB Biotech and then present a small selection of our portfolio holdings. Following this, we are thrilled to have Stéphane Bancel, CEO of Moderna, present Moderna, as well as give his vision for the company. With that, allow me to move to the slide deck and to the formal presentation. You see here the disclaimer that we are obliged to show, and with that, already to the next slide, where you see the quick rundown of today's presentation that I'd like to give. First of all, a quick overview over BB Biotech, following as said by the three examples. First, Relay, followed by CRISPR Therapeutics, and then just a quick introduction to Moderna. The company, BB Biotech, an investment company, was founded in 1993. So we are very proud to have now 27 years of track record, I would say a very successful one. And you see there a couple of facts and figures regarding BB Biotech being one of the leading investment companies in Europe with a focus on investing in the biotech industry. On the right-hand side, you see some figures and facts in terms of our daily liquidity, our annual dividend payout, as well as some cost factors and additional financial information. I am pleased to introduce you now the board of directors consisting of the 5 members. Let me start with the 3 gentlemen to the left that you all or most of you have already seen and heard about. Dr. Eric Hunziker, our chairman, joined the board in 2011. Then we have Clive Meanwell, who is member of the board since 2003. Thomas Fontplanter, who joined us in 2019. And then on the right-hand side, you see the 2 new board members that were elected in 2020, starting with Mats Krogsgaard Thompson, quite well known being the CSO at Novo Nordisk for over 20 years. And then Susan Galbraith, who joined us formally in October of this year. She's heading the oncology research and early development at AstraZeneca. The team that I'm proud to lead is what you see on the next slide. We have here 7 members that are part of the portfolio management team. Here as well, 1 new addition. Samuel Crozet joined us on October 1st. He's a bioinformatician. And then we have the 3 colleagues at the lower part that are colleagues from the investor relation team that are more than happy to follow up if you have further questions following this event. This is our representation of BB Biotech's investment strategy, or we can call it philosophy as well. It's our S-curve. Let me quickly walk you through this one. You see the time axis as well as the value or the growth axis as the y-axis. And we have our scope, or our focus actually in this red circle, meaning we invest when companies in the S-curve start to cut the red circle and start to move out. If the companies mature, we would divest and reinvest the company as an investment company at the lower part of the S-curve. Our focus is on small and mid-cap companies. We have a long-term investment strategy and a return on investment goal of 15%. The overall portfolio strategy is to have 5 to 8 core holdings, so meaning positions that are clearly overweight or higher weight, and then around 20 plus minus names that are smaller positions that we want to build or that are earlier in the S-curve as such. We are mostly US dollar denominated and can have between 5% cash and a 50% leverage as such. Over the last, I would say, decade, you see here we have broadened out not just the portfolio from a very concentrated approach to maximum 35 holdings, but we have broadened as well very much along the modalities or the drug technologies that we have within the industry. In 2010, that started off by investing into more complex antibodies and targeted toxin delivery, followed by, for example, antisense, that was Ionis in 2008 as well, then siRNA by Alnylam, followed by cell-based or investment in cell-based therapy being Juno and Kite. Then gene therapy with an Avexis example. You see there Moderna that we will present quickly as well today, that we did an investment in early 2018. And last but not least, the gene editing or the gene editing companies here represented, or in today's presentation represented by CRISPR Therapeutics. And last but not least, you see at the bottom to the right, Relay Therapeutics, a company in the small molecule world, but hopefully I can convince you why we're excited and thrilled to have Relay as our new position as of the third quarter of 2020. With that, allow me to present a quick breakdown of the portfolio. This is a new depiction. I think for most of our investors here on the left-hand side, the focus or the breakdown of the portfolio into these different drug modalities. The large part still being small molecule, followed then by actually the RNA and antibody companies, and then gene therapy, cell therapy, and protein companies making up the rest of the portfolio. And on the right-hand side, you see our usual split in terms of large, mid, and smaller cap companies. You see there by the third quarter, we had around 6% of the portfolio in the real large cap universe. That company was Vertex, or is still Vertex. Then we have in the dark red piece of the pie chart, actually, or the Kringle chart, the large or more than half of the portfolio within the mid-cap to larger mid-cap universe. And then around 40% in the below 5 billion mark, that where we apply actually and allocate most of the de novo capital, as we, for example, have done in 2020 by investing into 4 new companies in the first 9 months of 2020, all below the 5 billion mark as such. This is, I think, our result, which we are quite proud. On an even year-to-date basis, we could extend actually the very solid and great track record of BB Biotech once again. You see, or as I outlined before, we have a 15% return target in US dollar. That's a currency we invest into. You see there both share price and actually the portfolio, the net asset value are in line or very much along actually our long-term goals. And you see how actually the team together with the board could translate, as said, a portfolio that generates quite substantial mid and long-term outperformance for our shareholders as well. With that, I would like to close off already the formal quick rundown over BB Biotech as an investment company and move over to the first example of our portfolio companies that I would like to present today. The first one is Relay Therapeutics, a company focusing on small molecules, but we think a leader or an innovator in the rational drug design. And we have here on this slide actually the representation or the very quick rundown and summary page for Relay Therapeutics. The company is quite new in the stock market as well as in our portfolio because we joined or we invested into the company when the company did its IPO in July 2020, has already had quite a nice evolution in the stock market. And we think it's clear because of what the company has focused on and that's actually molecular dynamics simulation, meaning deep understanding of protein motion, connecting this with machine learning and, as I said, chemoinformatics methodologies to actually generate what we think is quite a unique pipeline. You see here 3 molecules in the lower chart that we have from the company. There's a SHP2 inhibitor, an FGFR2 inhibitor, which I'm going to quickly present to you, as well as a PI3K alpha program, still in preclinical stage. But as I said, we expect in 2021 then first clinical results actually for the 2 lead molecules. So what is actually intriguing or exciting for us to invest into Relay Therapeutics? And let me try to explain that slide quickly to you, because it's a combination of what the industry has done in the past, being the upper part in blue, so the experimental way how to do drug development, meaning in the wet lab, actually generating proteins, starting crystal structure, and then allow to run chemical libraries through being it cell-based assay or other functional assays, and through that generate drug leads and ultimately, obviously, drug candidates. And you see at the lower part, what is actually Relay adding to all of this? And it's their computational methodology, and I would say power as well. The key here is, first of all, on the left-hand side, to study protein motion, following then that they allow actually to screen these molecules in silico, meaning on the computer model, to have the chemical libraries tested out in these proteins that can move and function. And you will see this in one example in a couple of minutes. And then on the right-hand side, as I said, optimize the compound further so that we have an acceleration in terms of identification of lead molecules that move then into clinical trials. And to try to explain this more in pictures, I think that's often easier and actually more helpful. You see the same depiction of actually a fibroblast growth factor receptor 1 on the left-hand side in blue, and on the right-hand side, the FGFR2 molecule or protein, the structure of it. And the left part is actually how the industry has worked in the past, meaning with a crystal structure, a rigid structure that they looked at. And as you can see here, or hopefully you can understand, the molecules look extremely similar, very similar, and it's quite challenging actually to find binding sites that are unique for either one of the 2. So what has Relay done? They have, as said, studied the full protein function and actually the motion of it, and allow me to show this in a quick video. You see here on the right-hand side, actually the Relay approach. So on the left-hand side, FGFR1, where you see this upper arm move very quickly versus FGFR2 moving slowly. And now you see in red a small molecular binder come along, trying this pocket, and you will see that the FGFR1 is kicking the molecule out versus in FGFR2 it's stuck. And what it leads to is ultimately it blocks the signaling. So this electrical current that you see at the bottom part is actually the signaling of this protein. And I think it's, as I said, quite intriguing to see this. So let me show it once again, that you had time to study this. The red molecule is the binder or the inhibitory agent. And you see here the difference that on the right-hand side, the signaling is blocked. And that's how Relay could develop actually a very selective FGFR2 inhibitor that I said already has now moved into clinical studies. So we think a very unique approach, and we are looking forward, as said, to clinical results for Relay Therapeutics in 2021. The second example I try to explain here is CRISPR Therapeutics. We think a leading company in the field of genetic medicine. Why did we choose this? I can be quite frank, not just because it's a great technology. I think the technology got us well the reward by the 2 ladies that you see here, Emmanuelle Charpentier and Jennifer Duda, who received in early October actually the Nobel Prize for the technology, the CRISPR technology per se. And that's for multiple reasons. One is actually it revolutionizes the way life science is done in the lab. Secondly, we see it already applied in cancer therapies, meaning, for example, in cell-based therapies. And thirdly, there is expectation in the future that this may ultimately lead to cure for inherited diseases and severe disorders as such. We have 4 different technologies that are in the gene editing field. Top left, you see the meganucleases, followed by the zinc fingers, Then we have the talents as well. And let me focus on the lower right-hand part. That's the CRISPR-Cas9 molecule. That's actually the gene editing platform or technology that CRISPR Therapeutics uses. And allow me as well here to quickly introduce the company. We invested pretty much almost 2 years ago in the first quarter of 2019, when BB Biotech has done a more extensive investment cycle into genetic medicine. And you see the company had quite a good evolution, is already higher up in the market cap rank. But the reason or the rationale for us why we picked CRISPR Therapeutics was not just the lead on CRISPR-Cas9 technology, but as well because we like their approach to focus on so-called ex vivo application. You see there the company focused on the upper two parts of its pipeline on ex vivo application, meaning you take cells from patients into the lab, you genetically modify them, and reinfuse them as a genetic medicine back to the patient, whereas the lower part representing regenerative medicine and in vivo approaches that we think are higher risk, but very promising and with a lot of upside potential if they ultimately work. Here as well, a quick video how the CRISPR-Cas9 works. So you see there in orange, actually the CRISPR-Cas9, the enzyme or the molecule, the protein that binds a so-called guide RNA. And the combination of the two, you will see, it needs both, because I said the enzyme can only work if it has the guide RNA on top. And as soon as the two work together, they can actually start their real job. And the real job, or what they can actually achieve, is they can stream double-stranded DNA. And if they found the corresponding or the mirror image of the guide RNA, you will see here in the cartoon depicted, at that moment, the CRISPR or Cas9 will actually unwind the double-stranded DNA, lead to a so-called double-stranded break. So it really cuts the DNA into two pieces. And that kicks in the so-called repair mechanism that we have in all human cells. And often this non-homologous end joining leads actually to indel and inserts, and that leads to a change or a genetic alteration. And that's exactly what CRISPR has done for at least the two lead clinical programs that they have for beta thal and sickle cell. And I've quickly described this in short words at the bottom part. So what they do is they harvest, as I said, hematopoietic stem cells. They reactivate the fetal hemoglobin by turning the BCL11A gene off by this double-stranded cut and indel that get created. Then genetically, obviously, screen these cells if they're the right ones, reinfuse them, and you see ultimately the result of these patients. That's just a handful of patients. And let me actually start with the beta thal or the beta thalassemia patients. These patients have a misfunctional or dysfunctional hemoglobin because the beta chain doesn't work properly, and the patients suffer actually lifelong from severe anemia. And what you see on the right-hand side, very small depicted there, that they need a lot of actually transfusions per year to get their hemoglobin levels up and have a certain quality of life. And you see then on the left-hand side, actually with a single transplantation or stem cell transplantation, that over time, actually after a couple of months, the hemoglobin levels go north of 10 to 12 and almost 14 gram per deciliter, which is actually natural or normal levels, and B, that the fetal hemoglobin, the ones that, as I said, the CRISPR technology wanted to use, has actually achieved almost 100% exposure or expression, and we can actually foresee that these patients are cured for the rest of their life and don't need a further red blood cell or red blood cell transfusion anymore. And a similar depiction here for sickle cell disease. Similarly here, a dysfunctional hemoglobin. Actually, if the red blood cells are or the hemoglobin is deoxygenized, it leads that the red blood cells start to sickle, changes shape, and actually starts to block up and lead to a lot of pain and even to organ damage. You see this, the history of that patient, for example, on the right-hand side, once again, the veno-occlusive crisis. So every single red square here represents actually a severe clinical event. And at the lower part, you see that the patient requires quite often, or actually very repeat times of transfusion once again. And on the left-hand side here, you see that actually with the CRISPR technology, as I said, you can avoid or you can actually compete for the non-functional or the sickling hemoglobin. And around half of a set of hemoglobin is normal, and that's enough That's what you see on the right-hand side again, that the patient post actually stem cell transfusion had actually no more veno-occlusive crisis and didn't require any further red blood cell transfusion again. And this, a second example here in another disease where we can say the patients are ultimately cured with one-time treatment as such, and we think that's obviously, as I said, great news for the patient population. Last but not least, allow me already to move over to a very quick presentation of Moderna. You will hear much, much more about Moderna because obviously Stéphane Bancel will present about his company and can do so much, much better than me. But the rationale for us, first of all, so BB Biotech invested in first quarter 2018 when the company was private. We followed the company for quite some time before we participated, as I said, one of their last private investment rounds. And we were intrigued by the approach, but we want to see certain proof of concept. And here, two pictures that I would like to present that are outdated. They come from the S1 filing. And you see here, I said, a snippet of that. Starting on the top left, you see there a small ball that's actually a lipid nanoparticle that harbors so-called encoded messenger RNA. And it, as simple as it looks, it required the company actually to modify the messenger RNA to make it safer, to allow for repeat dosing, as well as obviously work a lot on the lipid nanoparticle to make it safe and functional. And what it allowed to do is actually, as I said, to transform this approach into what we think is a transformational therapeutic approach, because it renders human cells, cells of the patient, or in the case of a vaccine, it renders these cells into the machinery, or actually to result in endogenous expression apparatus for the function you transport by, as I said, the messenger RNA wrapped up in the lipid nanoparticle. And the company, already at private stage, had actually extensive pipeline, a broad and long list of preclinical assets, but already 10 of these candidates actually in clinical development. And that's what you see. That's still the stage of actually early 2018, and actually depicted here from the S1 filings, or then in late 2018. But you see here the 10 molecules. And our focus, and we had often debate as well with the company and the management team, our main focus was on what I've written here very simplistically in bold, two areas. The first one was infectious disease, the prophylactic vaccine, and I'm sure we'll hear much more of this today. And the second part was actually the systemic secreted intracellular and intracellular therapeutics for chronic dosing, meaning the repeat dose approach. And the company had other modalities. I'm not sure what we're all going to hear or what we have time for today, being in oncology, for example, in cancer vaccine, as well as cytokine expression, cytokine approaches, as well as in cardiovascular disease, where they even partnered up some of these assets depending as well on the risk early on. What has happened in the meantime, and this is now back to the formal slide, or actually the current status. So Moderna has, if you recall, our S-curve leapfrog pretty much from the earlier stage of a company all the way into a larger market cap company, but as well as into one that's soon going to have the first product in the market. And actually the leapfrog that happened was driven by rapid progress in the prophylactic vaccine space, mostly due to mRNA 1273, so the COVID vaccine. But you see here as well, it's a bit small depicted, that the company has already plans now to move the CMV vaccine as well into registrational trials. And I would say for us, an upside call on top, potentially even not being that recognized by Wall Street and the market because of all, obviously, the focus on the pandemic and the COVID-19 project, was that we have seen quite promising, actually, first data for the personalized cancer vaccine in combination with Merck's Keytruda, where we have seen, I think I said, promising responses in HPV-negative head and neck squamous cell carcinoma patients, and we'll be very interested, as I said, to follow up on this. And with that, I already want to close it out. Thank you for your attention from my side early on. And it's now my pleasure, and I said we are thrilled to have Stéphane Bancel from Moderna join our virtual Bio Day and present, as said, on Moderna as well as his vision for the company. Thanks so much. And with that, if we could switch over to the US.
Hey, Dan, can you hear me?
Yes, hi, Stéphane. Welcome.
Hey,
thanks so much for joining.
Thank you so much for having me. So if that's okay, Dan, I'm going to share a few slides and then we can go into Q&A.
Wonderful. Thank you.
Thank you so much. So again, good afternoon, everybody, and thank you for joining today. So before starting, let me remind you that I will be making forward-looking statements that investing in Moderna entails risk, and you can find the risk factors and these forward-looking statements on our website and also on the SEC website. So let me maybe start a bit where we started now close to 10 years ago with this notion that what would it mean for medicine if we could make medicines out of mRNA? So if you think about it, mRNA is an information molecule, and that's a piece that got me and us very excited to start the company. As I had to tell my wife, who's a photographer, when she asked me when I resigned from the CEO of Moderna role to join the company as employee number 2, what are the chances of this working? And at the time I told her, maybe 5% if we're lucky. But the piece that really drew our imagination at the time was the fact that in medicine, you never really had information molecule. And if you think about it, because mRNA codes for protein instruction, if you are able to get mRNA into a cell, as if you can see my mouse moving, get into the mRNA into the ribosome to make a protein, what was very clear to all of us is you could not only make secreted protein, like the biotech industry has done, that you can see in green for 50 years now. But what was very interesting and exciting, you could make intracellular protein or transmembrane protein. And if you look at it, around 2/3 of the protein coding for the DNA are proteins coding for transmembrane protein, intracellular protein. And that space is not really addressable using recombinant technology. And then, of course, we'll talk about vaccines in a minute. This is just human proteins. Then you can add proteins or viruses to make vaccines. So if you think about it, this was an incredible wide space of product opportunity. And you all know the value for patients and the value for shareholders of an approved molecule that is true differentiating product, kind of first-in-class medicine. The second piece that excited me, based on what I've learned, in my formative years working at Eli Lilly, is probability of success. So as you know, it's highly documented. The probability of a drug going from the labs to approval is very, very low. It depends by therapeutic area, it depends a bit by technology, but overall it's very, very low. And so one of our theses at the beginning, which I think is starting to play out in the data, was because you make the mRNA molecule from the same chemical structure, regardless if you make a vaccine for COVID, a vaccine against Zika, or vaccine against CMV, it is the same thing to a cell. The only difference is the order of the letters on the message. Like you will have zeros and ones coding software. As you all know, life is coded in 4 letters. It's just the order of the letters that change, but the chemical we use to make the mRNA, the manufacturing process to make the mRNA, the chemical we use to make the formulation to go around the mRNA, and the manufacturing process to make the formulation is the same, in the same equipment, made by the same people. And so if you think about it, what the cells see when you inject a vaccine from Moderna, it's the same thing regardless if it's CMV or Zika or COVID, the same thing. So what we said is that if one product is going to work in human and eventually get approved by the regulators for that application, there should be a lot of products, because then the only difference is the order of instruction on the messenger molecule. And so we believed that this will increase drastically the probability of technical success, because the raw material we use, that I think is often underappreciated by investors, is genetic information. If you think about it, the raw material of Moderna is either human genome, which has been sequenced, or the sequence of a genome of a virus, which costs around $5 to sequence. Those are the raw material we use. Then it's a technology that we've developed But what changes from product 1 to product 2 is only the order of the letters based on that instruction that is in nature. That's another very important piece. Most of the small molecules that have been developed do not exist in nature. In our case, if you think about it, the protein that is coded exists in nature. And that's, I think, is very, very important for lowering the risk of tox and increasing the risk, the probability of technical success. The third piece that excited me a lot is speed. Seeing the slowness of pharma just got me extremely frustrated over the years. You know, sometimes we had to wait a year or two between having a great molecule in an animal to being able to take it to the clinic, because we had to invent how to make at the FDA standard that molecule, because it was a new manufacturing process. Here again, the same process is used for every product. So once you figure out once, it's just copy and paste, and here you go again. So it was clear to us that if we invested in process development, robotics, and IT, we could really scale this company in a way that was extremely atypical for biotech and pharma. And the last piece, which was more the cherry on the cake, is you make mRNA without using cells, so you don't need the gigantic reactors that we all have seen in biotech companies that cost, you know, hundreds of millions or billions of dollars to build. You know, one of the last plants I was part of helping building at Lilly was an insulin plant for $1 billion just for making the insulin itself. And so this is much cheaper. It's a liquid phase, so it's in water, it's enzymatic. The cost of goods, we've always believed at commercial scale, will be similar to a small molecule, not a large molecule, a small molecule injectable product. So when you look at all those things together, we got very excited to say, look, let's take this low probability chance to build this company, because if it was going to work, it was going to impact patients for the next 50 years, 100 years. And that's the piece that you need to really understand about Moderna, is the team is extremely mission driven. We feel it's almost a duty to society to deliver on this technology for patients. And this I remind every new employee every Monday when I do the welcome introduction. And so as we thought about building Moderna, we spent a lot of time thinking about how do we build the best version of Moderna? Not the 1 or 2 product version of Moderna, but the best version of Moderna. And as we thought about all the things that could make us fail, we agreed with the team and the board that there were 4 main risks. If you go and read the SEC document, you see there's a lot of risk, like in any company. But those are the 4 risks that we talk about daily as a management team. Technology risk. Of course, if you go into a new technology, you can have a lot of technology risk, the usual unknown. Biology risk, which is, of course, how well some diseases are understood. Think about cancer, think about HIV, think about Alzheimer's. And there are some diseases are very well understood, like some rare genetic disease. If you know the mutation of a gene, you know how to go and fix. So it's a very low biology risk. Execution risk, and of course, as a company that had no product on the market, financing risk. So we spend a lot of time thinking about those 4 risks and how they intertwine together. And the conclusion we came up with the board and the management team early on is that we cannot take one drug into the clinic. We thought it was way too risky to take one drug and hope and pray that we had managed the technology risk and managed the biology risk. So what we ended up deciding to do, which is very similar to what you do as you manage financial assets, is we said we have to build a portfolio. We have to hedge our risk. And so we built basically 6 different technology applications that you see on the x-axis, from going intramuscular for infectious disease vaccine on the left in blue, to on the far right, injecting IV, a drug that is supposed to bring the mRNA into a liver cell to provide the instruction for kids that are a rare genetic disease, and a lot of things in the middle. So those colors, those verticals that we have are basically uncorrelated from a technology standpoint. They use most of the time a different formulation you put around the mRNA and a different injection route. And then on the biology risk, what we try to do is to start where we thought the biology risk was lowest, not where we thought the peak cells were the highest. We say, let's start to learn to walk before we run. And so if you look on the left at the bottom, we started with a pandemic flu strain to go into the clinic. And quite a lot of observers criticized us for doing that. And I told the team at the beginning, I said, I don't care about the NPV for our first product, because if it's $20 billion NPV times 0% chance of working, it will equal $0. So I say, what I care about the first product, it has to work. It has to work in a way that we know using an FDA approvable endpoint, is the technology working or not? Because if it's not, we have to go back to the drawing board. If it's working, we have to do more products in the clinic. And so that's really important. If you think about the company, one day, I was brainstorming with Gary Pisano, who is a professor at Harvard Business School, and he spent his entire life working with life science company. And he told me, it's very interesting the way you build the company, as he was looking at this matrix, and he and I were talking about it. It's like, the way I think about it now is on this x-axis, you guys look at like a typical biotech or pharma. To build a new technology, a new modality, it takes a lot of time. It's actually unknowable how long it's going to take you to get there. A lot of capital, and it's very risky. But on the y-axis, you guys look much more like a tech company, because you use information that's readily available. That, by the way, is free. It's the genetic information of human genome or a virus. And you plug that into your cassette in your technology, and you go again. Because all the products within a Koller are using exactly the same technology components. And I think that was a very interesting observation that he made, that indeed on the x-axis, things are risky and take time. This is basically the labor of the last 9 years to get those 6 modalities into development. But then scaling vertical, once you believe through clinical data that it's a de-risk and working safely, can scale very quickly. And we'll come back about that when we talk about COVID. An important thing also to know about Moderna, we don't think we are done with 6 verticals. We think there are another 10 plus verticals for us to develop. One of the verticals we're working on right now is actually delivering to the lung. We have a partnership with Vertex that is getting very close to getting the product into the clinic. And if that happens, we will add a vertical on the right as a 7th modality to bring mRNA via an aerosol through your mouth into your lung cells. And if that works, of course, then we can contemplate a lot of lung disease using that same technology. We are working also, we shared that on our Science Day 2 years ago, on how to bring mRNA injected IV into your white cells. This, we believe, would be very important for cancer, new application in cancer, and also autoimmune disease. So we keep investing in science to build new verticals, but I cannot tell you when the next one is going to come because it's science. So 2019 was a very important year for the company because 2 of our applications got clinical de-risking. So what we did basically when we ran the experiment of taking a lot of molecules into the clinic at around the same time was to let the clinical data guide our strategic decision. And we said, we're going to try those things. If we have positive clinical data, because mRNA is an information molecule, we'll scale many more of the same product in the same modality. If it doesn't work, we'll either shut it down, or if we think we have learned something interesting that we think as a technical solution, we might decide, it will all be based on facts and data, to take it back to the clinic. But we are very clear that we might shut down that application if we had negative clinical data. But those 2 articles highlighted in gray gave us positive clinical data last year. And so if you think about it, I believe that this was a very important year for the company because in my books, the company pivoted from being a company with 6 modalities that were in clinical exploration, because we only care about clinical data. Animal data are interesting, but clinical data we care about. And we moved to basically a company that has basically 2 different businesses. You have those core modalities that we believe have been technologically de-risked, where we said we're going to scale. And that's basically what we announced at JP Morgan earlier this year, that in those 2 modalities, we're going to come with a lot of products. And if you look at the company, before the COVID pandemic became intense and we focused a lot in it, we actually named 5 new development candidates in the first 60 days of a year in those 2 modalities. It was just us starting to scale up the company. And what we said is in those exploratory modalities that you see on the right, we will continue to be very disciplined to wait for clinical data and base decision on the clinical data, not to scale too fast or to scale too early. That was very important. So what we told investors early 2020 is we said, if you go back to 30,000 feet and think about Moderna and Moderna development as a company. No, we said the first 4 years of the company, up to December 2015, we were working on the technology, working on manufacturing process and manufacturing to be able to enter the clinic safely. Our first clinical trial started in December 2015. And then if you think about it, with this clinical readout into the core modalities, which I just spoke about at the end of 2019, we spent another roughly 4 years by accident to basically explore in the clinic 6 different modalities of mRNA. And so this was an important pivot at the end of last year, where 2 modalities became core, where we say we're going to scale them. And we say we're going to continue to investigate current modalities, and we're going to continue to invest in science to potentially create more modalities. And we told investors, based on where the assets are in the clinic now, it's around another 4 years to get to the first BLAs. By then, we'll get to generate cash, and then we'll scale the company pretty aggressively, because given we have a platform, if we're able to become sustainable cash flow-wise, we want to reinvest a lot of capital into the business to grow the company from a 20 product company to 30, 50 product company, because there's so many products we can do with messenger RNA that are not doable using other technology. So, of course, a new virus emerged at the beginning of the year, became clear to us toward the end of January that this wasn't getting to a pandemic. I can come back more to that realization and why in January, at the end of January, actually the week of the 20th, is when I realized this was going to be a global pandemic during the Q&A. But let me tell you a couple of things about what I think are the 2 big implications of our vaccine. As I'm sure most of you know, we announced final data of our phase 3 analysis yesterday, 94.1% efficacy. And what actually got me the most excited on Sunday is that we had 30 cases of severe disease out of the 196 total cases in the phase 3 study. And all 30 cases were on placebo. We had 0 case on active, which, as you can imagine, is a phenomenal breakthrough, because it means that if you get our vaccine, you have 94% chance to have no disease. And the 6% chance you have to have disease, it will be mild disease, it will not be severe disease. And if you think what's happening to the economy in terms of hospitalization, ICU, and death, and the impact it has on basically containment and so on, this is a big, big impact. And I think the 2 large implications of this is first, on the sales of these vaccines and the cash flow we would generate thanks to those sales. And I'll come back to this in a minute. And the other piece, of course, is the read across the vaccine platform. As I told you a few minutes ago, we believe that by application, by modality, there's incredible read across. The success of mRNA 1273, I believe, has no read across the rare disease or cancer, because it's a different technology. But it has incredible read across the vaccine. I'll come back to the implication in a minute. So what basically COVID has done to us, it has accelerated Moderna development from a development stage company to a commercial company. Instead of taking 3 to 4 years, it has taken actually a year for us to move commercial, because with the data we had yesterday, and we filed to US FDA yesterday by 4 p.m., the EUA was filed to the FDA. We also filed to Europe, to EMA, and we sent the data to Swiss Medics, to UK MHRA, to Canada, to Singapore, to Israel, and to WHO for low-income countries. So just a few facts about the vaccine. First, of course, you are aware of the speed at which we have been able to move. The sequence was put online by the Chinese authority on January 11. 2 days after, we had finished developing the vaccine. The research on the vaccine was done. The full sequence of a vaccine was finished. All the technical characteristics of the product were done and set in stone. That was 2 days, 48 hours. The product that we announced the data of yesterday was a product that was designed in a computer with never touching the virus in 48 hours by our team. And because of the work we had done on MERS, and because of the work we had done on the 9 other vaccines we put in the clinic before that one, we felt pretty confident it had a good chance of working. We started the clinical trial on March 16, To give you a sense of the speed, for the first SARS, it took Tony Fauci and his team 20 months to go from sequence to starting a clinical trial. Here it took 2 months. We started the phase 3 in July, and the interim data November 16, and final data of phase 3 yesterday. The FDA has scheduled its advisory board for December 17, so I'm expecting that within 24 to 72 hours after that advisory board, we should get an emergency use approval of mRNA 1273. The data in the clinic that I think was very encouraging this summer, that was published in the New England Journal of Medicine, that you can see here, is basically the neutralizing antibody data. The pink zone is basically the range of convalescent sera that the NIH, this is all coming from Dr. Fauci's team because they ran the phase 1. The NIH had collected from people that had been infected and had survived COVID, and you can see the range in pink. And what you see with the error bars is that the free age group, 18 to 55, in the middle, 56 to 70, and the right one, 71 and above, you see the level of neutralizing antibody. And I would want to draw your attention to 2 things that I think are really important. First, across age group, you see a very high level of neutralizing antibody at the top range of convalescent sera. And 2, if you look at the error bar, all participants in each age group made neutralizing antibody within a range of convalescent sera. And that is, I think, the power of mRNA that is massively misunderstood by a lot of people. With, let's say, adenovirus technology, if you go back to look at the clinical data, the error bar are very, very large. It's a bit like gene therapy, and it's not surprising because you use a virus as your vector to get into the human body. You see a very hit or miss. Some people respond very well. Most probably they've never been exposed to the virus before, and some people don't respond very well. In our case, all participants made a lot of neutralizing antibody, and you do not see waning with age, which is really, really important. If you look at other technology, including other mRNA vaccine, look at the data where you see a drop in the elderly data. Another big thing that our investment allowed is better storage and shipping condition than other mRNA vaccine. The other mRNA vaccine has to be stored at -70 Celsius. Our products can be stored at -20 degrees Celsius. So it's a regular freezer because they are products that have been approved at -20 for other pharmaceutical. All the big distributors of pharmaceuticals have in their warehouse freezers at -20 today. They do not need to go buy, install, and validate because this is all regulated industry. The freezer are -70. But then the big change is at fridge temperature. We are able for 30 days to have a product stored at fridge temperature, a regular fridge like you store insulin, or a fridge like you have in your house, and then 12 hours at room temperature for injection. This, we believe, is going to be a big differentiation. I've already got several health ministers calling in the last week or 2, asking for more product because they realize that managing the supply chain at -70 is going to be a massive headache if it's not well organized. So let's talk a bit about the sales and what it's going to mean for us next year. So what we've said is we have built the manufacturing capacity for between 500 million doses and 1 billion doses for 2021. In 2020, we should have around 20 million doses ready to ship as soon as we have regulatory approval. On the left, what you see is the deals that have been signed. So we've signed a deal with the US government for 100 million doses. The US government has options to buy up to 4 increments, 100 million at a time option. The EU has announced last week a partnership for 80 million doses with an option to add up to another 80 million doses. Japan with a partnership with Takeda, 50 million doses. Canada, 20 million doses. They still have an option to add another 36 million. And the UK and Switzerland and Israel and Qatar and many other countries. What does the price look like? In the early days, back in the spring, when we had no clinical data, just preclinical data and data on the other vaccines of Moderna, the price was at around $32 per dose. As we got the human data, the price moved to $37 per dose. This is for deals that are small deal in volume, think in the millions. For very large deal, like our biggest deal was the US government, We ended up doing a partnership at $25 per dose once you include the BARDA grant that we got from the US government. We have quite a number of deals with quite a number of countries that are in negotiation, including with COVAX, the facility set up by the WHO with Gavi, where we have proposed tier pricing to help low-income countries have access to this vaccine. So you can build your Excel spreadsheet with volume on the one side, with that range of $500 million to $1 billion, and the price to get a sense of it of what we might do next year. And this cash flow is going to, of course, be extremely transformative to Moderna. I'll come back to it in a minute. But what about the rest of the vaccine pipeline? So what I believe is because of this positive phase 3 data, this has increased the probability of technical success of all of our vaccines. Those in development, those in research that we've announced, like flu and HIV, and even those that are still in the lab that we have not announced yet, because they use exactly the same technology. Let me just pause a second on development program. Everybody is very interested by COVID-19, obviously. It's going to be a near-term catalyst. It's going to bring cash next year. Actually, already brought $1.3 billion of cash of prepayments this year by governments. But let me talk to me about the CMV vaccine. The CMV, cytomegalovirus, is a vaccine that causes the #1 cause of birth defect in the developed world. In Europe, in the US. In the US, we have 10,000 kids per year that are born with birth defect because their mom got unlucky. She was infected during pregnancy by CMV virus. We had positive phase 1, and then in September announced positive phase 2 data. The phase 3 is going to start next year. This is a product that is not partnered. Like mRNA-1273 for COVID, we have not partnered, so we keep 100% of the profits. This is a product that we anticipate CMV will have $2 to $5 billion annual peak sales. And if you think about vaccines, those are very long-tail products, as you can see from the sales of Prevnar, which is Pfizer's #1 product. Prevnar was launched in 2000. This is 20 years ago. And in last year, Prevnar was growing, still growing every year. Last year was a $6 billion product, 20 years after launch. A lot of investors do not appreciate vaccine. They think vaccine means a low selling price. It is true for Me Too vaccines. It is true for vaccine against tropical disease, but not about the vaccine that are on that page. CMV vaccine, RSV is a respiratory virus, no vaccine on the market. Like CMV, no vaccine on the market. HMPV is the third respiratory virus that's killing in Europe and in the US. PIV3 is the fourth respiratory virus that's killing people. EBV is Epstein-Barr virus. It's a virus driving mononucleosis, no vaccine on the market. It's a very complex herpes virus like CMV. It's using this very similar technology than CMV. Zika, we are doing as part of our corporate social responsibility. It's being funded by the US government. We're not doing it for profit. We're doing it because one day Zika is going to come back. I cannot tell you if it's next year or in 20 years, but when it's going to come back, it's going to hurt a lot of families, and the world needs to have an approved vaccine for Zika already. Even if the sequence change, we can within 30 days change the sequence and be ready to help. Seasonal flu. As you all know, the seasonal flu vaccine efficacy is pretty bad, in 30 to 60%. It's pretty bad in the elderly, which is, of course, the population at the greatest risk. Based on the great data we had in humans in the elderly in COVID, we think we have a chance to disrupt the seasonal flu business, and we're going to go at it pretty aggressively. And because we can combine different mRNA in the same vial, we could potentially combine a seasonal flu vaccine with a COVID boost or an RSV vaccine or more. You can think about a lot of combination. Just to give you a sense, our CMV vaccine, as we just spoke about, has 6 mRNA molecules in each vial. So we can combine mRNA to do very, very interesting product from a marketing standpoint. So that's for I think the read across around vaccine. And then you might ask yourself, but Stéphane, the vaccine you listed are the vaccines that can be done, and this is not a lot. It's actually just the opposite. Since 1980, more than 80 viruses have been discovered by clinicians and scientists around the world. There's only exist a approved vaccine against 2. Okay? So there's a lot of vaccines to be developed. And you see at the list at the bottom, if you can read it. If you cannot, please ask Dan or Chris the slide. They will be happy to share them with you. You see quite a number of viruses that were discovered before 1980. And those in red still do not have a vaccine on the market like CMV, Zika, or EBV. So there's still a lot of vaccines for us to do to increase the pipeline. So could I see a Moderna pretty soon that could have 10, 15, 20 vaccines in the pipeline? I think it's highly possible. So let me maybe close on that last slide before taking question. I believe 2021 is going to be the most important inflection year in the company history. And why do I think that? So let's think first about the beginning of 2020. We entered this crisis in a strong position. $1.26 billion of cash, a diverse portfolio across 6 modalities, a total of 20 development candidates, many more candidates in the labs. We made sustained investment in the platform. The piece you need to know about Moderna, we are very, very long-term driven. We think it's still early days of this technology. We think the S-curve is still on the low part of a ramp of the S-curve. So we keep investing aggressively in science, and we're going to keep doing so. We have a very large IP portfolio. We have sustained investment also in process development. As I told you, the difference between our vaccine and BioNTech Pfizer vaccine for storage, it's investment in process development that we made over the year. And we have our own manufacturing plant. So we started the year in a good place. Now let's think about where we're going to end up 21. So I'm asking you to think and imagine where Moderna is going to be around 13 months from now. So first, we're going to have a very large cash balance. I think a pretty unique cash balance in the history of a biotech company our age. We already had $4 billion of cash at the end of September. In Q3, we generated $1 billion of cash flow from operation during the quarter. So think about it. I think we are in a unique position. The US government has sponsored only 2 mRNA companies. That's it. There's no space for any more. And of course, Moderna is one of those 2. We got $1 billion paid by the US government to pay the entire development plan of mRNA 1273. But then the piece that differentiates us from BioNTech is that we have not partnered the product. So think about this. The Moderna team was able to pull the development of this product by itself. And at the finish line, we are 1 week behind Pfizer. Pfizer is 100 times the size of Moderna. They have more people in communication or lawyers than I have scientists. And we're going to keep all the profits. We're not doing a 50-50 profit share like they are doing. We're keeping all the profits for the future benefit of Moderna shareholder to be able to invest in more product, to scale the company, to create more return and more value into the pipeline and into commercial product. As I said, we are very close to getting a new modality in the lung on top of the 6. I could see a world where by the end of 21, 13 months from now, we don't have 20, but more closer to 30 development candidates. We're going to continue to invest in science. We're going to continue to build a bigger IP portfolio. We're going to continue to invest in process development. And thanks to what we built for COVID, including our partnership with Lonza, we will have by the end of the year, next year, a manufacturing infrastructure able to do 1 billion doses per year, which is really important. So I think the next 12 months are really important. Moderna is going to scale. If you think about the company, while we have always been well capitalized, we have always been needing more cash to invest in more product. We have a platform. We invested in science to develop the technology. We invested in manufacturing. In Norwood, we can make up to 100 new development candidates in the clinical trial each year. That's how the plant has been designed, up to 100. So what is the fuel that we have been missing so far is enough cash to scale. So 1273 has not only accelerated the development of a company to move into a commercial company, it is getting us very soon to a place where we're going to be sustainable by generating our own cash flow and not needing to raise capital on the market by selling stock. And it's going to give us a very strong balance sheet so that we can invest to scale Moderna, so we can continue to build the best possible version of Moderna in the next 5 years, 10 years, 20 years. So with this done, I turn it back over to you. I'm going to take the slides off and be happy to take any question.
Thank you so much, Stéphane, and thanks for sharing the presentation. I think that was a great overview. As outlined in the introduction, we have set an extensive number of investors on this call, and I think if I hear from my colleagues, we got quite a list of questions. So if you don't mind to stay a couple more minutes on the call, Stéphane, that would be great. I don't know if my colleagues from Investor Relations would start actually with the question roster.
Stéphane, thank you very much also from our side. We have received many questions focused, of course, on the COVID-19 vaccine. The first question would be, how long can antibodies be detected after administration of the vaccine? How long will the immunization last?
Yes, that's a great question. So we don't know yet. That's one of the frustrating things about developing products so fast because of a pandemic. We are measuring that as we go. We're going to be able to share very soon, and I mean very soon, data from the phase 1 study post-boost, because the phase 1 started in March. We anticipate, it should be at least a year or more, but we have to get that data to be sure. I also expect it might look different into an older person versus a younger person. So the frequency of a boost might need to be different based on age. But again, we have to be a bit patient, wait for the clinical data. I expect in the next days, we'll learn a bit more from the phase 1. But what I expect is in the coming months, we will be, again, very forthcoming to communicate what we learn and to publish it in peer-reviewed journal.
Great, thanks so much. I think the second question, what I hear as well to you, so makes my life easy to steer the discussion. I think we have time now for the second question. That works.
Stéphane, could you please give us some color on the manufacturing and the costs of the COVID-19 vaccine of such an enormous scale?
Yes, so I'm going to give you a bit of manufacturing picture. I won't talk to you about cost. We have not disclosed cost for obvious competitive reason. And because of Reg FD, I, of course, cannot do that on this call. On manufacturing, so basically, the manufacturing process is as follows. We start by making a plasmid, so basically a piece of DNA that is used as a template for our product. Then using enzyme in reactors made of water, we make the mRNA molecule. Then when it's made, we add the cap, think about it like the nose of a molecule. When the mRNA molecule is made, we have to purify it to remove all the byproducts that are coming from the manufacturing process itself. And then when the mRNA molecule is made, we basically mix it with lipids. So think about it's fat that we have developed, for which actually these were invented by our team. We have IP on that composition of matter. And that becomes basically a formulated mRNA. So it's mRNA molecules with kind of little balls of fats called lipid nanoparticle, LNPs. And then when those are made in big plastic bags, think 30, 40 liters of vaccine, they are put into vials. Usually it's 1 dose per vial for regular commercial products. For the pandemic, after talking to the authorities around the world, we decided to put 10 doses per vial just to increase the throughput, because there was not 14 billion of vial capacity sitting idle in the industry waiting for a pandemic. We still need, as an industry, to fill product for cancer, diabetes product, and so on. And so we thought it was a nice way to be able to get more out of the manufacturing of vials. So that's kind of the manufacturing process as a whole. If you look at what we did in January, it took us 42 days to go from locking down the product to shipping the product after quality control, including 2 weeks of quality control on the back end. So as you can see, the cycle time is pretty quick, and we can then, as soon as the product is made, go back, make another lot, and keep going.
Thanks for that. That's, as I said, very impressive as all the rest we heard today. I think there is at least one more follow-up, if I'm informed. Maria Grazia.
Yes, Stéphane, when will the vaccine become available here in Europe and in Switzerland? And when do you expect the FDA approval?
Yes, so let me pull those things apart, because those are 3 different actually questions. So on the US front, we filed yesterday our EUA to the FDA. We announced that on social media at the end of the day. We announced in the morning it was going that day, but just after 4 p.m., the FDA confirmed receipt of the EUA. And we've said that the FDA has informed us that our advisory committee in the US is likely to be on December 17. And what I would expect is within 24 to 72 hours after that meeting that we should hear from the FDA. I expect, and I believe this should be a positive answer given on the one hand, the medical need and the pandemic, on the other hand, the clinical data that we have been sharing. Of course, FDA is going to get all the detail of the data. But that's roughly when I think we'll get US approval. I would say between the 18th and the 21st of December. And then we'll basically have the trucks coming to pick the product at the factory by pallets of vaccines. And what the Operation Warp Speed in the US, which is the US government, has prepared for and has confirmed again recently publicly, is that they believe they have all the infrastructure set up to start vaccinating Americans within 24 hours from when we hear from FDA that FDA has given us approval. So that's for the US. In Europe, we have also filed yesterday with EMA, which is the regulatory agency for Europe. This morning, EMA has issued a press release saying that they confirmed that they've received the request for conditional approval from Moderna for mRNA 1273. And they indicated in their letter of a press release, that they are scheduling their advisory committee January 12 or earlier. So of course, we were hoping that it was going to be December, but as you can appreciate, this is not in our control. So it seems for Europe, based on what we learned this morning from the EMA, that we could have an advisory committee on January 12. And I would again expect within 24 to 72 hours after that committee an approval in Europe. As soon as we have approval, same thing as the US, we'll be ready to ship product. We have already made products for Europe out of our European supply chain, and they will be ready to go. In Switzerland, we have also yesterday submitted the data to Swissmedic. We have been in contact with Swissmedic for now quite a while, providing to them all the pieces of the file that we could send to them, the preclinical animal model, the phase 1 and phase 2 data, but also all the manufacturing process, because a lot of people don't always appreciate that manufacturing and quality of a physical product itself is, of course, as important as the efficacy data or the safety data. Those are the three pieces of information that the regulators really look at. And so for now, several months, we have been filing regularly as we generated new manufacturing data, the data to the regulators. So Swissmedic has also all the pieces together. What we basically did yesterday is we submitted the data at the same time to the US FDA, to Canada, to Europe, to the UK, to MHRA in the UK, to Swissmedic, to Israel, to Singapore, and to WHO. So all those agencies are starting at the same time on the clock speed, and we'll see when they get approval. I don't have more granularity in Switzerland as to when could we anticipate an approval. Is it this year, or is it more like Europe early in the new year? What I can tell you is something, as soon as we get the green light from Switzerland, our European supply chain will be ready to send product to Switzerland. I have to say, because I know we have a lot of Swiss investors, that the Swiss government has been extremely proactive. They ordered their first mRNA product back in the summer. As they get more data, they ordered more products. And I can tell you, the volume have not been disclosed, so I need, of course, to keep confidentiality. But there is a very good coverage of the Swiss population with the Moderna vaccine that has been accomplished by the Swiss government to protect the Swiss population.
Right. I'm looking for further update, even from a personal reason or from a personal stance. We just heard yesterday actually from the Bundesrat that I said they consider actually an early 2021 launch, at least to the media, whatever that's worth, but that was the latest update yesterday, at least in context of Switzerland. I think there's one last question to Stéphane, if I did it correctly, if you don't mind.
Stéphane, we have a question about long-term side effects. Did you get already some feedback from general public?
Yes, that's a good question. So a few pieces of context. So we have been doing vaccine clinical trial for now 5 years, or almost 5 years. The first trial started actually in Germany on December 9, 2015. And we have tried 9 vaccines before the COVID vaccine with, as I said, the same technology. So it's interesting to know that we've dosed a couple thousand people with our vaccine around the world, and we have not observed long-term safety side effects. On mRNA 1273, the product has been in humans since March 16. We have not observed long-term side effects. It's important to know in vaccines, and this data was discussed at the advisory committee of FDA that happened, I think in November, is that in vaccines, when you look at all the vaccines from all the manufacturers across all vaccine technologies, 99% of long-term side effects actually happen within 6 weeks after the boost of a vaccine. So that's reassuring because the FDA has asked that all the manufacturers wait for at least 8 weeks because of that 6 weeks historical data to put a little bit more room for safety, that we will not able to submit an EUA. We passed that 8 weeks mark, 2-month mark, post or boost on the second half of November, which is why we were able to submit the data to FDA for EUA approval yesterday, as per the FDA guidelines. So that's for the macro things. Now, for those of you that have science background, let me share a couple of things with you. The mRNA molecule that we make has around 6 hours half-life in your cells. By 48 hours, you cannot find it anymore. And we've done incredible science in vivo to show in human cells, in vitro, and in animal, including monkeys, that after 48 hours, the mRNA molecule is degraded inside your cells. It's cut in little pieces for all the little nucleotide, each letter of life cut from the mRNA molecule. We have shown and published it on our website that the mRNA molecule does not get into your nucleus. This is one of the many reasons I loved mRNA since the beginning. I was always worried about safety, having seen, at Lilly, just drug failing after drug failing after drug failing in the clinic because of tox. So that's for the mRNA molecule itself. Now let's talk about the lipid. The lipid that we develop has a few hour half-life. So same thing, once it brings and delivers the mRNA into your cells, it basically falls apart. It was designed by our chemists to basically be eternalized by the enzymes that are inside your cells. So if you think about it, within 24 to 48 hours after injection, none of the product we injected in your body is still there as an active molecule. And actually, if you look at the side effects that are not severe, that have been published in the New England Journal of Medicine, you will observe that they all appear within 24 to 48 hours after injection, that they look like a typical vaccination. You have local side effects, like pain at the site of injection. Your arm, is a bit swelled for a day or so and hurts. Some people have a bit of redness around the site of a needle track, like sometimes you see with a vaccine. All those go away by themselves within a couple of days without any medication. And the systemic side effects are very also typical of what you see for approved vaccine. Some people have a tiny bit of fever. It's less than 2% of people. Some people have a bit of headache. Some people have a bit of fatigue, especially in the elderly group. The same, it goes by itself with no medication within a day or two. So it's a pretty mild side effect profile, consistent with approved vaccine. No severe side effect has been observed so far. And as I said, we have been at it from the technology for years on this vaccine since March, and 30,000 people have been tested in that study and are extremely closely followed. They all have an app on their phone, and they are asked every day if you have side effects from the vaccine.
And if I may add, I said we will get the outcome data as well soon. So I think all the way through the program, we had I would say, a congrats to you and the team. Always a very high transparency in terms of where you stood about the progress, both in terms of efficacy as well as safety. I think that's, as the question probably tended to, is very important to convince, I said, the broad population towards this new technology, which I think, I said, results are obviously, very encouraging, extremely positive, and I think we are all, as such, impressed by this. I think there's one last question, actually, towards BB Biotech.
Daniel, yes, a question for you. BB Biotech has seen a strong appreciation this year despite COVID and the related lockdowns. What were the drivers besides Moderna?
Yes, thanks for the question. I would say, I said, as usual in biotech, we have seen actually the best performing or the best returns, at least in our investments, in companies that have shown, I would say, transformative clinical data. And the good news was we had quite a few of these in 2020. Started off with mavacamten for myocardia, that ultimately led as well to the transaction or the takeover of Bristol Myers Squibb. We had, for example, argenx with very positive phase 3 data for efgartigimod, which we think will be approved in 2021 for AMG. And I said, for example, even smaller companies with early clinical results, such as Koller, who I think showed quite intriguing proof of concept data as an add-on therapy to Spinraza for different patient populations with actually spinal muscular atrophy. And obviously everything was, if I can use the word, trumped or overrun pretty much by all the news and headlines that we had around COVID-19 and the pandemic. And I said for us, it was impressive to follow Moderna and all the progress. I think that's absolutely astonishing what Stéphane and team have achieved. I think we as shareholders obviously got the reward as well. And normally I don't do that, but we can announce now that Moderna is our largest portfolio holding as of the last couple of days. So we see, as I said, we think a long-term holding into the right technology such as Moderna can be of big reward if you take the time, study and analyze companies, and then hopefully, as I said, pick the winners such as a Moderna. And with that, Stéphane, I think I can thank you from the entire team, the board, all of our participants, that you took so much time in these busy times. We know there's a lot going on, and there will be a lot more going on at the company. Wish you all the best, being it the upscaling into an ultra-large scale manufacturing, the shipment, all the updates, and obviously the formal approvals as such. With that, thanks so much for taking the time, and we'll be more than happy to follow Moderna for the years to come. And with that, I want to thank as well our attendees to this virtual buy day. Thank you so much for taking the time to listening to, as I said, the quick intro to BB Biotech, and to the highlight, I think, of today clearly being, as I said, the Moderna update from Stéphane. Thanks everyone, and wishing you a good day.